Imagine proteins traveling through a pH highway until they reach their sweet spot, their isoelectric point, where they stop in perfect neutrality. That's isoelectric focusing, IEF, one of the most precise ways to separate amino acids, peptides, and proteins in molecular biology. Now let's recap. All amino acids share two key functional groups: the amino group, NH3+ when protonated, and the carboxyl group, COO- when deprotonated. At any given pH, these groups can gain or lose protons, changing the overall net charge of the molecule. Recall that protonation is just making a positive charge, and deprotonation is making a negative charge through the loss or addition of protons. At a low pH, less than or equal to 1, both the amino and carboxyl groups are protonated, so the net charge is +1. At a high pH, greater than or equal to 12, both are deprotonated, so the net charge is equal to -1. Recall that pKa is the negative log of Ka. pKa is the pH at which a group is 50% protonated. And Pi, the isoelectric point, is pKa1 plus pKa2 divided by 2, just an average for simple amino acids. So at the isoelectric point, Pi, the amino acid exists as a zwitterion, one group positive, one group negative, with net zero charge. Remember that the isoelectric point is just the pH where the net charge is 0. Now there are differences among amino acid types and the Pi values and how they're calculated. For neutral R groups, such as alanine, shown above, Pi is roughly 5 to 6, because it's the average of the pKa, which is approximately 2.2 and 9.5 for the amino group and carboxyl group. For amino acids with basic R groups, on the other hand, such as arginine or lysine, you might see a case like this. Arginine has a pKa1 of approximately 2, pKa2 for NH3+, of approximately 9.0, and a pKa for the R group of roughly 12. There's a neutral state between 9 and 12, so the Pi is roughly 9 plus 12 divided by 2, averaging out to about 10.5. Basic R groups typically have a Pi around 9 to 11 because additional basic side chain pKa is included. On the other hand, for amino acids with acidic R groups, Pi is roughly 3 to 4. It's lower due to the extra acidic side chain. Consider this example with aspartic acid. pKa1 is roughly 2, and pKa of the R group is roughly 4. The neutral form between those would be a Pi averaging 2 plus 4 divided by 2, about 3. All of this is to predict whether an amino acid will carry positive or negative charge at a given physiological pH. And now things get exciting, looking at how isoelectric focusing actually works. Isoelectric focusing uses a gel embedded with a stable pH gradient, ranging from acidic, a pH of about 3, to basic, pH of about 10 or more. We apply an electric field across the gel. This pH gradient is just an increasing pH across the gel, and the electric field that we apply drives molecules toward opposite charges. Proteins in this setup will migrate based on their net charge, positive toward cathode, negative toward anode. As protein moves, surrounding pH changes, and when it hits its Pi, net charge becomes 0 and movement stops. That's where it focuses, creating tight band separation. Focusing is just the accumulation that happens at Pi. At that point, there's no further migration. So, what makes IEF so incredibly powerful? Well, this method achieves extremely high resolution, separating proteins differing by only a single charge. That's critical for analyzing isoforms, slightly modified versions of the same protein, or analyzing proteins with post-translational changes, such as phosphorylation. And once separated, these focused proteins can be extracted and analyzed by other techniques, like mass spectrometry or 2D gel electrophoresis, for even more granularity. Let's do a little example to see what we can glean from this kind of test. You see the protein blue band would be at about pi of 10.1. It would stop near the basic end, suggesting plenty of amino acids are basic. And the protein at the yellow band would be at a pi of about 5.6, stopping earlier, closer to an acidic region, which suggests that acidic residue content is in the sample. So what are some common questions? Well, why do we average pKa values rather than just select one? Because to get net zero charge, the amounts of positive and negative groups must be an exact balance, which mathematically occurs at the midpoint. Why do externally changing conditions affect pI? Well, extreme environments, such as high salt or strong denaturants, can shift protein conformation slightly, but the intrinsic pI remains consistent for that sequence. Can proteins with multiple ionizable residues have multiple pIs? No. They only have one net pI, but their migration zone can appear broader if many residues are ionizable. So, in summary, amino acid charge shifts with pH via protonation or deprotonation. Each amino acid or protein has a specific pI, isoelectric point. In isoelectric focusing, molecules migrate until they reach net zero charge, forming tight bands. This is how scientists sort proteins precisely, based on composition differences. If you learned how proteins travel and stop at their pI and enjoyed the video, please hit the like button and share your molecule of interest. Want to learn more about how this is used in 2D protein separation or biomarker discovery? Subscribe and let me know. Thanks for watching.